Thank you, Andy. Another fabulous presentation. Now, our last presenter that I've got the pleasure of introducing is Professor Ian Beveridge from the University of Melbourne. Ian worked as a research scientist in Switzerland and Townsville, and then took up a teaching post in veterinary parasitology in Adelaide, and later at the University of Melbourne. And he did that in 1989 and has been there ever since. Now, although paid to do other things, Ian's main interest has been parasites of wildlife, and Ian will enlighten us now on how to really make the most of a road trip in Australia. Thank you very much, Ian.
Shouldn't give it away. Well, thank you, and good evening, everybody. I guess very few people in this room, I suspect very few people in this room, even realised that 2010 was the United Nations Year for Biodiversity. And I'm pretty certain that virtually nobody knows that this is the current decade for biodiversity. If you visit these United Nations websites, there are hundreds of wonderful proposals for conserving biodiversity. They run on for pages and pages and pages, and they're all very, very laudable. When we think about biodiversity, the pictures on this slide are really the sort of things you tend to think about. Pretty flowers like Banksias, not such pretty plants like seaweeds, a variety of marine invertebrates like tunicates and sea stars and sea pens, and even furry cuddly things like wallabies and kangaroos and wombats. And that's fair enough. But in all of these discussions about biodiversity, the one important component that is overlooked are the parasites, because all of these animals have parasites. And it's been estimated, I think guesstimate is the better term for it, that perhaps parasites, if you lump them all together, may constitute between 30 and 50% of the entire biodiversity present on Earth. And I've just put up a few examples here. I've put up three different orders of tapeworms from the intestines of different sorts of vertebrates, a couple of orders of nematodes. I'm sure the trematode people will be absolutely incensed that I don't have a trematode there, but never mind. And there's just examples of external parasites. This is the mite that causes scabies in humans, and I've put up a picture of a tick. I mean, there are lots and lots of different types of parasites. So it will be really interesting to know how biodiverse parasites are, and the estimates that we have are really very, very crude indeed. Now, the estimates depend on what assumptions you make about at the start, and essentially how you define a parasite, which, as we saw earlier, could be contentious. But the estimates range between 30 and 50%. I don't know how the 30% figure comes up. The 50% figure is relatively easy to explain. If you assume that each animal on the face of the Earth has one parasite, then obviously 50% of the life forms are going to be parasites. That's very, very simple. Very straightforward. But perhaps it's an underestimate. A lot of hosts that we know about actually have more than one parasite, so perhaps even these estimates are grossly misrepresent what's truly there. So I thought I'd just take one example this evening, a local example. Take kangaroos, because they're familiar to all of us, and they're quite diverse. There are about 70 species of kangaroos, wallabies and rat kangaroos in Australia and Papua New Guinea, and they're reputed to be wormy. So I thought I'd investigate this and have a look at biodiversity in parasites of kangaroos. You can see the diversity we have. We've got nice little things that you can hold in your hand, like musky rat kangaroos, and I've just put up examples of some of the other different types of kangaroos and wallabies. I don't think a lot of people don't realise the diversity of kangaroos that exist, and I've put about 70 species because every second year the number creeps up a bit as people realise that the species they thought was extinct isn't, or they find a new species. So, and it is going to increase a bit more in the foreseeable future. So they're an interesting group to work with. The other reason is that they're quite easy to work with. We can access them quite easily. They're shot commercially in most states, including Tasmania. We can get them from culls, and what you see up here are two postgraduate students processing animals that have been culled by farmers who've decided that they've got too many kangaroos on their property, so they get a permit to shoot them. And the other option is road kills here. Tasmania is noted for many things, and one of the things it's noted for is the abundance of road kills. And these actually, if they're fresh enough, provide a valuable resource for parasitologists researching parasites of native wildlife. And if you're travelling in, this obviously isn't Tasmania, but if you're travelling in less populated areas, you can collect your samples off roadsides, and then the local motel can very easily be converted into a laboratory. You carry a couple of microscopes with you and set them up on the breakfast bench, and you can process your samples in the local motel, although it's probably a good idea to be discreet about what you're doing. Collecting roadkills, you have to be a little bit selective. Sometimes if some larger transport vehicles have actually run the animal over, there's not much to be retrieved from a carcass of this sort. So we've got a wide diversity of animals we can collect from. Generally speaking, they're easy to collect, and we can sort of process the material quite readily. When you do open up a kangaroo's stomach, this is what you're likely to see. This is an agile wallaby from northern Tasmania, and what you see is a whole lot of what look like noodles. If the animal's fresh enough and still warm, when you open the stomach, a lot of them are still wriggling. What you can see there is that they soon attract blowflies, which are a ubiquitous part of the Australian countryside. But if you actually look more closely, beyond these noodle-sized nematodes, what you see is that perhaps up here you can see that in fact there are lots and lots of much smaller worms. So you get the idea that these kangaroos have lots of worms in their stomach. And if you're a parasitologist, what you do, well, you count them. Now, for the non-parasitologists here, you don't literally count every single worm. You use a dilution technique whereby you take a sample and then extrapolate from the sample. But I just pulled these figures out from the literature. These are some of the common kangaroos, like the two grey kangaroos and the red kangaroo. They weigh about, on average, 40 kilograms, and these are the average numbers of worms in a kangaroo. So an average red kangaroo has nearly 60,000 worms. And what you can see is that the range is enormous. So some grey kangaroos have up to 500,000 worms. And when you go down the scale to little rock wallabies that only weigh nearly 8 kilograms, you've still got averages of 23,000 worms. So your perceptions are quite correct. There are lots of worms present. If you go one step further and actually put these things under a microscope, you discover that not only are there lots of worms there, but there are lots of different species present. Now, these are just a few examples. These are scanning electron micrographs, and these are just pictures taken under a standard microscope, and I think they're 10,000 times more attractive than Giardia, but we won't tell Andrew about that. But there are the message is that not only are there lots of worms, there are lots of species. So the question is, we're interested in biodiversity. How many species are there? So this is the current state of play on just the group of worms that occurs in the stomachs and occurs in these big numbers. So, right, we've got 70 species of kangaroos. We, at the moment, have 38 genera of nematodes and essentially 300 species named of these particular nematodes. Now, if you look in the literature, you'll see that horses and zebras, equids, are held up as an example of particularly wormy animals in the large intestine in the case of horses. If you look at horses, equids here, there are only 5 species still extant. We've got 13 genera and 51 species, so the kangaroos beat them hands down. And the other group that's used are elephants and rhinoceroses, and again we've got 8 host species, 4 genera of nematodes and 48 species of nematodes. So the kangaroos win all round. And I've put a little asterisk there simply because this is a conservative estimate. And it's conservative because, first of all, not all hosts have been examined, and we have this problem with sibling species that I'll explain. Now, you don't have to worry about the names on this slide. You just have to look at the colours. First problem is we actually haven't looked at all of the species of kangaroos available. I've put about 50 up on here. The ones we've looked at for parasites are in white. The ones we haven't looked at are in orange, and you can see that the orange ones far outnumber the white ones. So we've got a long way to go. So the first thing we've got to do is actually look at all the remaining kangaroos. Now, this might be getting a little bit complicated, but the other problem we have is with sibling species. Now, nematodes are relatively simple organisms. They've got nice, pretty mouth parts, but the rest of the body is pretty uniform. This is an example of rock wallabies. Whoops, sorry. in rock wallabies, we have a series of species that occur up the east coast of Queensland, closely related species of rock wallaby. And in them, we've got 3 species of nematodes that we can clearly identify under the microscope. It's quite simple. You can just pick these species apart looking at them under a light microscope. The problem is, when you collect material of these 3 species from various rock wallabies and actually look at their DNA sequences, the story is much more complicated. Now, I realise this diagram is a bit difficult to follow, but it's actually fairly straightforward. So what we've done here is we've taken one of these species of nematodes, or what we call a species, from 5 different rock wallaby species, and lo and behold, we get 5 completely different DNA sequences. We've done the same here with 6 rock wallaby species, and we've got 6 completely different DNA sequences, and down here we've got 5. So we've looked at 3 species of nematode, and suddenly we've got 15 different DNA sequences. Are these different species? They probably are. The DNA sequences are quite significant. They're not closely related to one another, and if you use a phylogenetic approach to this, then you are obliged to refer to them as distinct species. So what we think is that once we start looking at these nematodes with more advanced techniques, such as DNA sequencing data, we're going to uncover far greater diversity than we ever suspected existed. So that's why I say our current state of knowledge provides us with considerable underestimates of biodiversity. So that's one of the problems with these estimates. I don't think for many groups of organisms we really have any good understanding of how biodiverse a group is. Now, I said that the United Nations were very, very concerned about the conservation of biodiversity. And that's wonderful when you're talking about wombats and kangaroos, but it does present a few problems when you come to parasites. Do people really want to conserve parasites? And we have a lovely local example here in the Tasmanian devil. The Tasmanian devil is host to a number of parasites. The Tasmanian devil is threatened. We hope it doesn't become extinct, but if it were to, then there's a series of parasites there that would become extinct along with the Tasmanian devil. Now, the ones with asterisks I've pulled out simply because these do also occur in quolls. So they may be able to maintain their life cycle in quolls, but the numbers in quolls are very, very much lower. The others occur only in Tasmanian devils and will become extinct if the devil becomes extinct. Now, this poses a couple of practical problems. As you know, there are reservoir populations of devils being set up in mainland zoos. They're caught in Tasmania, or have been caught in Tasmania, from areas free of facial tumour disease. They then have health checks, and then they're transported to the mainland. And presumably the vet's doing these health checks, wanting to make sure the devils are in the best of health, treat them with an anhelmintic, and kill these potentially endangered nematodes. Now, what should they be doing? Should they be treating the devils before they go over to the mainland, or should they be attempting to conserve this biodiversity? I don't have an answer, but I just raised the question. I've put one parasite here in yellow, Dasyurotenia robusta, because this is one area in which Tasmania stands out. Several years ago, due to the work of David Ovendorf, I think about 15 years ago, David was surveying devils from all around the state for Trichinella pseudospiralis, and as a sideline was looking at other parasites and realised that this parasite of the Tasmanian devil was actually very rare 15 years ago. Tasmania put it on its threatened species list, and I think that was a historic event. I think Tasmania at the time was the only place in the world that had a parasite on its threatened species list. Now, with threatened species, generally, there is a recovery program put into place. Well, of course, no recovery program was put into place for poor old Dasyurotenia. It was restricted to the northeast, and the northeast is one of the areas that has been most severely affected by devil tumour disease. So it may be that Tasmania holds the record, or will hold the record, of having the first species of parasite on a threatened species list to actually have become extinct, because no remedial activity has been undertaken. So that's the converse side of biodiversity. So I'll finish up by thanking you for your attention and just pointing out that perhaps when you think about biodiversity and the conservation of biodiversity, you might give at least a brief thought to the parasites as well as to the hosts. Thank you. I stay here.